Our presentation is over the diversity of soil invertebrates within different growth areas. I'm Hannah Kochis.
I'm Natalie Nicholas.
I'm Emily Fisher.
And I'm Devin Weckerly.
Introduction. The definition of soil fauna are the animal members of a soil community. Soil ecosystem plays a key role in cycling nutrients throughout the biosphere. It supports decomposing organisms, and it also contains nitrogen-fixing bacteria. Most common organisms can be found in the phylum Nematoda, Annelida, and Arthropoda. Soil organisms like to live in the dark, moist, semi-aquatic environments. They use touch and smell for orientation. Most have water permeable skin, and physiologically restricted to semi-aquatic environments. And they also use negative phototaxis, which is the tendency to move away from light.
Alright, so our article that we found to support our hypothesis was called The Energy Use in Animal Abundance in Litter and Soil Communities, and it was done by Timothy Nahan. And he said that the higher the temperature, the more abundant and diverse the soil invertebrates are going to be. So we thought that the new growth site has smaller trees, so the temperature would be higher, so therefore there would be a greater abundance and diversity. And the old growth site has larger trees, so the temperatures are going to be lower. Our objective was to compare the soil invertebrates in the two different sites in the new growth forest and the old growth forest. And we thought that the new growth forest would be more diverse than the old because the trees were smaller, so there would be a higher temperature in the soil to allow for more abundance and diversity.
Methods were, our location was the University of Mount Union Nature Center in Lyons, Ohio. And the old growth forest and the new growth forest were the two sites that we sampled from. The old growth composed mostly of American beech trees and the larger trees because of the longer growth period. Whereas in the new growth forest, it was composed mostly of maples and smaller trees, so smaller growth period. In the old growth forest, there was also larger trees, which equals less sunshine, and it's more hilly, so there's less retention of water. Whereas in the new growth forest, there's less trees, more sunshine, and it used to be farmland, so it's a flatter terrain where there's an increased retention of moisture. For our sample, we filled a 1-gallon Ziploc bag loosely with leaf litter we found from the two different sites. The setup, we used a Berlese funnel to extract and capture the soil invertebrates. As you can see, the lamp was set over the soil, was set up over the soil sample and placed in a large funnel. The invertebrates traveled away from the light and to the funnel where they dropped into the alcohol. In identifying the invertebrates, we used the Quick Heat Soil Dwelling Invertebrates Guide, and we studied each of the samples that we pulled out. When calculating the diversity, we used the Shannon-Weiner indices to compare the diversities of the two different sites, where you can see H' and H max to determine the diversity.
Our results for the new growth forest and the old growth forest were a little bit different. As you can see, in the old growth forest, we had a total of 9 different species, with the columbella being the most diverse. We actually found an estimated of over 700 because every time we laid out, they literally covered the surface. Whereas as opposed to the new growth forest, the columbella was also the most abundant group, but there was only about 102, and we only had 5 different types of species in the new growth forest. When we were looking at the results of the old growth forest and the new growth forest, when we were comparing the H' and the H max, we found that the new growth forest was actually more diverse because the difference between the H max and H prime was only 0.49272, whereas in the old growth forest, the H max minus the H prime was 0.8039. And we do think that there was a little bit of a skewed number because of how many columbella we found in the old growth forest, being over 700. And the dominant groups in the old growth site were the columbella, the acarina, the urania, and the coleoptera. And the dominant groups in the new growth site were the columbella, the urania, the coleoptera, and the lepidoptera. And like I said before, the new growth forest was more diverse with a difference of only 0.4972, and the old growth forest was less diverse with a difference of 0.8039.
So in conclusion, our hypothesis was the new growth site would be more diverse than the old growth site. Our data did support our hypothesis. And referring back to the article of Meehan, the new growth forest site would be more abundant and diverse because of the lower tree density leading to more sunlight, which led to higher temperatures. And then, as Devin said, the overabundance of Columbella and the old growth forest may have skewed our results for diversity.